So I'm going to tell you about my own program at Harwell, which is type 2 diabetes and obesity, and the functional analysis of genes involved in type 2 diabetes and obesity. So I'll start with a bit of background on diabetes. There are two types of diabetes. We work on type 2. Type 1 is an autoimmune disease where the beta cells of the islet, the cells that secrete insulin, are destroyed by the immune system. In type 2 diabetes, there are defects in insulin secretion, but also in insulin action, so that you need more insulin to have the same effect on glucose uptake into insulin-sensitive tissue. So over time, you may develop insulin resistance. Your beta cells will increase insulin secretion to compensate for that. But as time goes on, as the insulin resistance worsens, there can be a failure of the beta cells to compensate, and then that results in high blood glucose, which is diabetes. So it's a very common disease. Over 366 million people are affected worldwide, and this is predicted to rise quite substantially. 25.8 million people in the US. In the UK, it's estimated around 3 million people have type 2 diabetes, but there's another 860,000 or so who are undiagnosed, who aren't receiving treatment. Life expectancy is reduced by diabetes by about 10 years. And as an example, in the UK, the healthcare budget spent on diabetes and diabetes-related complications is around £27 million a day. I mean, these are figures from 2012, so it's going up all the time. So there are significant implications for mortality, morbidity, and for the cost to the infrastructure for treating and supporting patients. Age is a factor in type 2 diabetes, so it's less common in young people, but in the US, people aged over 65 years or older, about 10.9 million people in this age group have diabetes. Age is a risk factor. There's an increasing prevalence in younger people due to increasing obesity rates. So there are increases in that younger age group, which is a particular worry. So diabetes is high blood glucose, but the thing that's a problem is the complications that high blood glucose over long periods of time cause. So it increases your risk of cardiovascular disease, and it's in over half patients with type 2 diabetes, it's the main cause of death. Eye disease, it increases your risk of going blind. It's the leading cause of blindness in the UK. It's the leading cause of end-stage renal disease, where you require dialysis or kidney transplant. It affects peripheral vasculature, so you get effects that damage to peripheral tissues, which can lead to limb amputations. There's nerve damage, which partly contributes to this limb amputation problem. And nerve damage is present in up to half of patients. Depression, sexual dysfunction. So there are a lot of morbidities associated with diabetes over the long term. So as I've alluded to, obesity is a risk factor for type 2 diabetes. A large proportion of patients are obese, but not all patients are obese. You can get diabetes and still look relatively lean, although you may have more visceral fat, which may not be obvious, other than on an MRI scan. And it's a sliding scale of risk, so the more obese you are, the more risk you are, and the risk can be up to 40-fold. BMIs over 30 kilograms, a particular particular risk, so your risk increases exponentially once you reach this point. And it's estimated that the risk of developing diabetes could be substantially reduced by increasing activity levels. So if you can increase activity levels in the population, you'll see a drop in diabetes. As I've already said, age is a risk. The population that you're from is also a risk factor, so diabetes risk varies with different ethnic groups. People of an Indian background have a higher risk than, for example, people from China. So this points towards genetics being a risk factor, varying risk in different populations. So a classic example of a high-risk population are Native Americans and the Pima Indians in particular have a 50% rate of diabetes. Other risk factors for diabetes are fetal growth and intrauterine environment. So low birth weight is associated with diabetes in later life, thinness at birth, small size in relation to the placenta. So intrauterine conditions are important in diabetes. So is there a genetic basis to type 2 diabetes? It's clearly familial. There's an increased risk to first-degree relatives, 3 or 4 times higher than the general population. So if you have a first-degree relative who has diabetes, you're at 3 or 4 times greater risk. There's greater concordance among genetically identical homozygotic twins, between 50 and 90%, depending on the study that you read. And it's lower, obviously, in dizygotic twins who are not genetically identical, around 37%. Incidence varies between different ethnic groups, as I've said. There is a relationship between incidence and the degree of genetic admixture. So if you look at populations that are mixing and estimate the degree of admixture, you can see that corresponds to risk. Segregation analysis suggests complex genetic control. There are rare and monogenic forms of diabetes, subtypes of diabetes, and there are common genetic polymorphisms that have been mapped by genome-wide association studies, and there are more than 90 of these genes so far. So these all point towards a genetic component. The GWAS loci aren't necessarily very predictive of your risk. You have to type a large number of genes before you get anywhere near having some predictive value. But the value of type 2 of these GWAS approaches is that these are genuine risk factors, and they do contribute to disease at the population level, but they tell you something novel about the biology. So clearly there's a genetic basis, but there are risk factors that interact with the genetics to determine your overall risk. So you may inherit risk factors, but you may never get diabetes if you're not exposed to some of the risk factors. So why study diabetes? Well, we need better treatments. There's a limited range of drugs, metformin, sulfonylureas, thiazolidinediones, which aren't so much used. More recent treatments based on incretins, which potentiate insulin secretion and compounds that inhibit the degradation of incretins. We need better treatment so we can reduce the complications of diabetes and improve quality of life. So the aim is to get at new genes and pathways, understand the biology better, and hopefully find new therapeutic targets and strategies. So the particular focus of my group is on genome-wide association study candidate genes. So the value, as I've alluded to, for GWAS genes is that they're real, well-replicated human risk alleles. They may be small effect, but they're real. They're mostly novel genes, novel biology, therapeutics, and therefore novel therapeutic opportunities. And in investigating the biology underlying these genes, they provide targets for patient studies. So you could recruit patients on the basis of their genotype to follow mechanism in patients. So our approach is to test candidate genes in these GWAS loci, so that these GWAS loci contain a number of genes, and it isn't clear usually which gene is contributing to the risk, which gene or genes. It may be more than one gene within a locus that's mapped in the human population. And our aim is to test candidate genes using multiple alleles in mouse models, and to apply to those models comprehensive metabolic phenotyping, those models that can then provide tissues and organs for mechanistic studies. So, you know, there are 90 or so loci, so we're selecting loci based on data from human geneticists, our collaborators, who are involved directly in these genome-wide association studies and are doing follow-up studies, such as sequencing patient groups and doing fine mapping. And we're choosing genes that are supported by other human data, where we can, through our collaborations, potentially get access to patient cohorts that are recalled by genotype. So the aim is to do detailed mechanistic studies on these genes to understand how they function, to investigate their therapeutic potential. So we have a source of genes on the left there from genome-wide association studies. There are genes coming from other sources as well, but the main focus is on GWAS. There are lots of alleles that we can have from these genes. So there's the IMPC alleles that you've already heard about. We can make overexpression alleles. We can use point mutations from E and U. And with new technologies like CRISPR-Cas9, we can engineer alleles. So we can make a variety of alleles for the genes that we're interested in and apply metabolic phenotyping and get models. So I'm going to illustrate this with the work that we've done on the fat mass and obesity gene, FTO, the human GWAS gene. In 2007, this was first published, and I've quoted there are a number of papers that year, but this is one of the main ones, the Frayling Science paper. This was an association with introns, with SNPs within intron 1 of the fat mass and obesity gene. 16% of adults are homozygous for the risk allele, and they weigh 3 kilograms more. FTO and another variant in another gene implicated, well, directly involved in obesity account for about 2% of the variance in adult BMI in the population. The effects of FTO can be seen from 7 years of age in terms of body BMI, and it's associated quite clearly in the human population with increased energy intake, so food intake. And this is Andrew Hattersley and Tim Frayling who are key to this work. So as I said, the associated SNPs are in the first intron of this gene, FTO. There are other genes in the region, and if you read the literature, you'll see various studies that point to the involvement of other genes, RP-GRPRL, and IRX3, which I won't really talk about, but I can answer questions on those if you want later on. So what about FTO? So FTO is the gene that we've selected as a candidate. It's a 2-oxyglutarate-dependent nucleic acid demethylase. Other family members are implicated in DNA repair, fatty acid metabolism, post-translational modifications. It's nuclear localized. And the initial studies showed that it had a preference for single-stranded RNA and DNA, and particularly for 3-methylthymidine, for demethylating 3-methylthymidine. But it's emerged over the last 2 or 3 years that perhaps the most important substrate for FTO is M6A methylation, and various studies have shown clustering of M6A methylation of RNA around stop codons at the 5' end of mRNAs and around splice sites. So it may be involved in RNA regulation, and I'll come back to this briefly later on. So this is an unusual function. It's hard to see how this would be linked to obesity. It's expressed widely. So the SNPs, one of the things you can do when you know you've got an association is to look at expression of the genes around your associated interval. And people have done this for FTO. They've looked in various tissues, and it's a bit of a mixed story as to whether or not expression increases or decreases. So in human obesity, so in obese patients in subcutaneous white adipose tissue, you see an increased expression of FTO. This is regardless of genotype. This is just looking at the expression of FTO in different tissues. In the brain, in the hypothalamus, a number of studies in rats and mice have also been a bit contradictory in that fasting rats showed an increase of expression and starved mice showed a reduced expression. But these studies all vary in the amount of time that the mice or the rats are starved for. And in this case, this really was starvation. It wasn't just fasting overnight. It was much more extreme than that. So it seems from these studies that FTO may well be responding to energy demand. People have looked by genotype, and there are reports that there are no differences, that there's no correlation with the risk SNPs and FTO expression in peripheral blood lymphocytes, because those are the easy tissues to get. But other studies have found that expression varies according to the genotype. So this Berubler paper in the European Journal of Human Genetics showed a 1.38-fold increase in transcripts carrying the risk allele. And similarly, in this Cara JCI paper, they also found an increase in FTO abundance with the homozygous risk allele. So this is distinguishing alleles, again, in peripheral blood, which may not be the right tissue. We don't know what... We have some ideas now about which are the key tissues, but obviously we can't access the hypothalamus very easily for RNA. So there are a number of alleles. There's the human GWAS, obviously. There is some evidence for increased expression with genotype, with the risk allele, and obviously this is associated with increased BMI, fat mass. There is a recessive point mutation that disrupts the 2-oxyglutarate activity in a single family, and this causes growth retardation, malformations, and unfortunately lethality. There's been a study where heterozygous mutations have been looked for in FTO in lean and obese individuals, and there was no correlation with the heterozygous mutations and leanness or obesity. There was the first report of a mouse null allele, so this again was recessive, where there's no protein, so this is a knockout mutation from Fisher. And these mice were lean, showed growth retardation and lethality. And I'll show you some of our data on this model as well. We have our own model. We published on a point mutation, which disrupted dimerization of the FTO protein, and this resulted in leanness. So much milder allele than this knockout, so a hypermorph potentially. Or a dominant negative, actually, because this reduced dimerization. And we have an overexpression model, which I'll show you as well, where we have increased expression and dose-dependent obesity, supporting FTO as the gene. So I'll go through some of these models and then come back to where I think we are. So this is the FTO global germline knockout, which was worked by Chris Church, a student in my lab, and Fiona McMurray, also a student. This shows the FTO locus, exon 2, 3, and 4 of the locus. Chris targeted this locus and inserted LOX-P sites either side of exon 3 so that we could delete exon 3 using a Cre recombinase. In making these constructs in ES cells, he had to use a selectable marker, which is here, neomycin, and that's flanked by some FRET sites. So once he'd made the cells and made the line and got the mice, he removed that selection cassette, which was necessary in the targeting steps, but we don't really want it there in the subsequent experiments. So he was able to then generate a conditional allele. So we have this allele where we have chromosome 3 flanked by LOX-P sites, and we can remove those LOX-P sites. We can remove that exon by recombination between the LOX-P sites using Cre recombinase. So in the initial experiment, we carried out recombination using a globally expressed beta-actin Cre, so Cre that's expressed in all cells, including germ cells. So we were able to generate germline knockout mice by this approach. So the mice I'm going to show you look like this. They don't have exon 3. So our germline knockout had reduced size, so they were shorter, and reduced body weight. So you can see in the blue line, hopefully this is not too faint. No, it's okay. The blue line, the heavy blue line is the knockout. And the wild-type littermates are in the lighter blue. So you can see they reduced body weight. They had reduced fat mass, as you can see here. Lean mass was also reduced. They had improved glucose tolerance, but we saw no effect on food intake. When we looked at energy expenditure using indirect calorimetry, so these mice are lighter, and one possibility is that they may not be eating as much as wild types. When we looked at energy expenditure, so this is measuring oxygen consumption, carbon dioxide breathed out, and calculating energy expenditure. We saw no difference in energy expenditure. It matters how, and the RER is calculated using these values and some other values so that you can work out, so you can estimate substrate utilization in these animals. So there's a trend towards fat metabolism in these animals. So we saw no difference in energy expenditure. There have been reports in the literature in knockouts that there is an increase in energy expenditure, but it matters how you normalize this data. So when you're measuring energy expenditure, obviously you have to take account of the fact that the mice are smaller, because a larger mouse is going to have to expend more energy to live and to move around than a small mouse. So you have to take some account of the fact that the mice are not the same weight or the same body composition. So when you look at the raw energy expenditure, you can see there's a correlation with lean mass. And if you try to take account of the lean mass by just dividing by lean mass, you end up with a significant difference in energy expenditure. But all you've done is divide by lean mass. And what you really need to do is to adjust for variation in lean mass using linear regression. So you make a correction for the differences in lean mass. If you just divide by a lower number, you're going to get a bigger number, which is what happens when you just divide by lean mass here. So it looks like the lower mass mice have a higher energy expenditure because you've just divided by their mass. So it's important to correct this sort of data properly. And these are all normalization and techniques that have been worked out in humans. So calorimetry has been done in humans for a very long time. And the mouse needs to adhere to those same sorts of standards. And otherwise the literature fills up with data that may not be reproducible. So the knockout mice have reduced size, so there may be developmental defect of some sort, reduced weight, reduced fat mass, reduced viability. So I didn't mention that, but they're not particularly viable. We lose a certain number before weaning. And there's unchanged energy expenditure relative to lean mass. So FTO is clearly necessary in early life, and it affects growth and fat mass. We wanted to avoid the lethality and ask what happens when you remove FTO in the adult. So FTO is present in these experiments that I'm about to show you during embryonic development, so we should get away from the smallness and the lethality, the early perinatal lethality. So this is an experiment that Fiona did, and this is all published and open access, so you can download those papers. So in this experiment, we wanted to knock it out later on in life. So we used an inducible Cre. So this is the same process and technology as I showed you before. We're deleting that exon 3 that's flanked by LOX-P sites using a Cre recombinase. But in this case, we have a Cre recombinase on a ubiquitin promoter, so it's expressed mostly everywhere, although perhaps not in beta cells. And we can turn this on using tamoxifen, which binds to the ER receptor. So we treat the mice with tamoxifen and for several days, and this induces the Cre. We get recombination, and then your gene is deleted then in those mice. And the hope then was to overcome the poor survival and growth retardation. So we were thinking we might see something similar to that ENU allele, which I briefly told you about. So I'm just going to show you two groups of mice. We had other groups. It's important to have the right controls, but I'm just going to show you the two main groups, and we had a number of other control groups. So the homozygous floxed mice treated with tamoxifen, carrying the Cre, and a vehicle-treated group. So these mice are identical. They all carry the homozygous allele with the flox elements on around exon 3, and they all carry the Cre. But in one, we turn on the Cre with tamoxifen, and in the other, we just treat them with the vehicle. So here's the treatment here in the little, here about 6 weeks. So we treat them for a week with tamoxifen. And then you can see that the dark blue mice here, the tamoxifen-treated mice, then diverge in body weight. It's known that tamoxifen has an effect on food intake, but in this experiment, we're treating for a week, and then the tamoxifen is gone. Whereas the vehicle-treated mice follow this profile. So, and these mice are approximately the same size, same length. So there's no difference in length. So at week 9, there wasn't much difference in fat mass, slightly increased fat mass. By week 20, however, very surprisingly, which was not the result we expected, was that there is increased fat mass in these mice. Lean mass is reduced at both time points. So the dark blue dots are the tamoxifen-treated mice. So these mice have reduced lean mass. So they seem to not put on lean mass, and they gain fat mass. So there may be a redistribution of substrate metabolites to fat and away from lean tissue. So we saw no effect on food intake. Food intake is very difficult to measure accurately because ideally you'd like to house mice individually and measure what they're eating, which is what you do for the calorimetry. You house a mouse individually, but it's quite stressful for a mouse to be housed individually, so you may get stress effects. So if your model is more susceptible to stress, you may see a difference in food intake just because it won't eat, because it's stressed by the environment. So we have been looking at better ways of measuring food intake, and we've started to measure food intake from mice that are held in pairs. So we have 2 mice of the same genotype, so there isn't quite the same single housing issue. And we put them in a cage and hold them for a week together in those pairs and measure food intake over the week by weighing the food pellets, but you have to search the cage for other bits of food. And so it's not as clear, it's not as clean perhaps as doing it in a classic metabolic cage, but that's a very stressful environment with a wire mesh floor so that food doesn't get lost, and a very open environment. So you need to think about how, when you look at food intake data, you need to think about how is it being collected. So we saw no effect on energy expenditure and indirect calorimetry, although again we saw reduced RER, which indicates more fat metabolism. So we got much improved viability in these adult knockouts, increased fat, decreased lean, no effect on energy expenditure, increased protein and fat metabolism. So FTO clearly regulates metabolism and body composition, but it's a more complex relationship than perhaps we initially thought. So these mice that I've been describing, well, the adult onset had increased fat mass, but the knockouts, the lack of expression mice, were lean. The reported associations with BMI with the risk allele are increased fat mass. So the question was, what happens if you overexpress FTO? So we decided to, Chris Church in my group just decided to overexpress FTO and see what happened to the mice. So to test the hypothesis that the risk alleles in the human GWAS studies increase expression of FTO in a key tissue or in multiple tissues, and that this causes the increase in BMI. So testing this idea, we used FTO overexpression. So the ROSA 26 locus is convenient because you can target a gene into that locus and know that you haven't disrupted any other part of the genome. So if you were to, you could just randomly insert a bit of DNA with a promoter on it to express your cDNA, but you might create mutations doing that by inserting your gene into the middle of something else that's really important. But the advantage of using the ROSA 26 locus is that we can insert things there without doing any damage. So he specifically inserted this construct. So this has a chicken actin promoter on it. It's got, again, a selectable marker and a stop codon. So it won't express from this chicken actin promoter until we take the stop out. And as you can see, we've got some LOXP sites. There's the FTO cDNA, and we've also got a GFP marker with an independent ribosome entry site. So we can follow the expression of this cDNA, or at least of this construct rather than the cDNA. So again, using Cre recombinase, we can delete the stop codon and the neo and switch this cDNA on. So we can switch this on whenever we like. So the experiment I'm going to show you, we switched it on globally again with the beta-actin Cre, but we've got the option to go on and switch it on in other tissues, which is what we're doing at the moment, in particular in tissues that we're interested in. Chris looked at the expression of this. So taking the stop out by beta-actin Cre recombinase, he saw an increase in the expression of FTO. And you can see it quantified here at the RNA level. So perhaps not surprisingly, in muscle, we saw the highest expression. So this is the copy number of FTO. So 2 copies is a wild type, because obviously there are 2 copies of the endogenous gene. 3 copies is a heterozygote for this ROSA 26 locus. And 4 copies means we've got 2 copies of this, 2 copies of the endogenous gene. So in the 4 copies, you can see quite a nice increased expression. You can see an increased expression in all the tissues that he looked at, but this is on an actin promoter, so muscle was the strongest. Body weight increased, so you can see the FTO4 mice here. This is male and female data, female on the left and male on the right. And you can see an increase in body weight here in the green line compared to mice that just have 2 endogenous copies. So overexpressing FTO increased body weight. This is on a standard diet over time. On a high-fat diet, this is also true. So you see an increase in body weight. And you can see quite a substantial increase when you open up the mice in expansion of fat pads. It's a dose-dependent increase. So we go from 2 copies to 3 copies to 4 copies here if we just look at the male on the high-fat diet. So you can see there's a dose-dependent increase in fat mass. So adding 1 extra copy increased body weight, and adding another copy, so you had 4 copies increased body weight. This is an effect on fat mass. So again, we've got male and female data. This is lean mass on this side, so there's minimal effect on lean mass. But when you look at fat mass, you can see that fat mass is increased. So if we go, this is on a high-fat diet again. So if we just look at the males, high-fat diet, 2 copies, so they're the wild types, as it were. 3 copies, an increase in fat mass. 4 copies, a bigger increase in fat mass. When you look at visceral fat depots and weigh them, you can see an increase in the weight of these fat depots when you've got 4 copies on the standard and high-fat diet. When you look at the area of fat cells, we also saw an increase in fat cell size. When we measured food intake, we saw an increase in food intake. So food intake on a standard and a high-fat diet on both diets, 3 copies and 4 copies of FTO increases food intake over 2 copies. And this is 10 weeks and 19 weeks, so the same story. So in summary, we found increased body weight, increased fat mass, hyperphagia, dyslipidemia, impaired glucose tolerance on the high-fat diet, no differences in metabolic rate when you normalize for lean mass, no differences in activity detected by wheel running or circadian period measurements. So we have these different models of FTO. We have a knockout that has reduced body weight, adult onset knockout, which have reduced body weight, but surprisingly increased fat mass at the expense of lean mass. We have a knockout where there's reduced body weight, reduced fat mass, reduced lean mass. We had an ENU allele, which I haven't shown you, which had a similar phenotype, body weight, reduced fat mass. And these FTO4 mice, the overexpression mice, where we get an increase in fat mass. We also, which another experiment which I haven't shown you, but which is in that PLOS genetics paper, is a medial-basal hypothalamic knockout using a virus targeting vector to knock out the, or to deliver Cre to knock out the FTO just in those parts of the brain where we saw only a moderate reduce in body weight. So FTO phenotype is not directly linked to energy expenditure. It has a role in early life viability and early growth. It has a role in body composition and metabolism. Other hypothalamic brain areas may well be involved, but other tissues are clearly involved. So it has a complex role both in brain controlling food intake, or this may be an indirect effect on brain that controls food intake, and in controlling peripheral metabolism, as I showed you, a switch to more protein and fat metabolism. So in the final bit of my talk, in the last few minutes, I'll just show you some stuff that's in press at the moment in Nature Communications. And again, this will be open access, so you'll be able to download it. I'm just going to give you a very brief overview of this. So prolonged high-fat diet results in both hypertrophy and hyperplasia. So if we look at these fat cells here, if we put them on a high-fat diet for 5 weeks, you get the fat cells fill up with fat. If you keep feeding them a high-fat diet, not only do they fill up with fat, but you get new cells as well. So you get hypertrophy and hyperplasia. And as I showed you earlier on, our high-fat fed FTO4 mice showed an increase in the size of adipocytes and also in the number of adipocytes. So when we counted them, the number was increased in the FTO4 overexpression mice. And we saw that, in actual fact, if you look at the distribution of the sizes of these adipocytes, we found there were more small adipocytes as well. Taking out visceral fat depots from the mouse and looking at gene expression, we could see, looking at visceral fat depots, we could see an increase in key genes in adipogenesis, such as PPA or gamma in the overexpression mice, but in subcutaneous fat, we didn't see these differences. So, and it's interesting to note that subcutaneous fat doesn't undergo de novo adipogenesis when you put mice on a high-fat diet. So this appears to be a visceral fat trait. So looking at the stages in generation of adipose depots, mesenchymal stem cells undergo a commitment proliferation step, and there are 2 obligatory mitoses, the mitotic clonal expansion steps, to generate pre-adipocytes, which then differentiate and fill with lipid. So we were interested in looking at where, if given the effects on fat mass, where FTO fits in with this, and given the differences I showed you in cell size and cell number. So when you take out primary adipocytes from FTO overexpression mice that have been fed off a high-fat diet and differentiate them in culture and then stain them with oil red O, which sticks to lipids, you can see in wild type there's some nice differentiation here and accumulation of lipid droplets. But in the overexpression mice, you see a very substantial increase in the amount of lipid. So FTO overexpression seems to promote adipogenesis. And again, looking at these primary adipocytes 10 days post-differentiation, you can see strong expression of these genes, PPR gamma, perilipin, which is involved in storage of fat, and fatty acid binding protein, which is involved in metabolism. So FTO overexpression promotes adipogenesis, and we see this in mouse embryo fibroblasts as well. So if you take fibroblasts, mouse embryo fibroblasts out of embryos and do this experiment, you see the same result, because you can differentiate these into fat cells. But if you do it with a knockout, you get the opposite picture. So FTO promotes adipogenesis, while FTO knockout inhibits adipogenesis in vitro. And FTO overexpression is associated with increased adipocyte number and adipogenic gene expression in visceral fat. It promotes adipogenesis in primary adipocytes, and we think this is we think it has its impact on the mitotic clonal expansion step. So, as I said, this is in press at the moment. Another paper came out while our paper was being reviewed from Zhao et al., and they report that FTO impacts splicing of this factor called RUNX1T1. There's a short and long isoform, and FTO demethylation promotes the short isoform, which is pro-adipogenic. So that gives you a sort of functional link now with FTO. It's known M6A demethylase activity, adipogenesis, and the process of determining fat depot size and function. So I've almost finished now. So that's that's where we are with the FTO story. So taking, I've illustrated, hopefully this illustrates to you the power of taking novel genes, making alleles, doing metabolic phenotyping, and then going forward to mechanistic studies. There's still a lot more work to do on FTO to understand its function and its role in early adipogenesis and how that fits with the adult phenotypes, the adult fat mass phenotypes. So can early events in adipocyte differentiation impact on adult BMI? So this work was started by Chris Church, continued by Fiona McMurray, and Sam Labber has picked up the baton from them and is doing the work on adipocyte, early events in adipocyte differentiation. And this is a collaboration with Fran Ashcroft in DPAG, and Diane and Myrta are also working on this adipocyte project. So thanks very much.